In this lesson, we're going to explore one of the most fundamental questions in biology. Why did evolution choose cells as the basic unit of life? And we'll see how physics and mathematics constrain biological design. The answer lies in a fundamental principle of physics called the surface area to volume ratio. This is one of the most important scaling laws in biology. Let's start simple. Consider a spherical cell with radius r. We can write down two key equations that govern its behavior. The surface area is 4 pi r squared. This is where the cell exchanges materials with its environment. The volume is 4/3 pi r cubed. This is where all the cellular machinery and metabolism occurs. Now here's the crucial ratio. Surface area to volume equals 3 over r. Notice this is inversely proportional to the radius. This tells us something profound. As the cell gets bigger, its surface area to volume ratio decreases. This creates a fundamental constraint. Look at this comparison. A small cell with radius 1 micrometer has an A over V ratio of 3. But if we make the cell 10 times larger, the ratio drops to 0.3. That's 10 times less surface area per unit volume. You might be wondering, why does this ratio matter so much for life? Let me explain the biological consequences. All cells need to constantly exchange materials with their environment. Nutrients come in, waste products go out, oxygen diffuses in, carbon dioxide diffuses out. The metabolic rate of a cell scales with its volume, because all the biochemical reactions happen throughout the entire cell interior. But the exchange rate scales with surface area, because materials can only cross the membrane at the surface. Metabolic demand is proportional to r cubed, because it depends on volume. But exchange capacity is proportional to r squared, because it depends on surface area. The ratio of exchange capacity to metabolic demand is proportional to 1 over r. As cells get bigger, they can't supply their interior fast enough. Let's work through a concrete numerical example with oxygen diffusion to see how limiting this becomes. Consider a typical bacterial cell with a radius of about 1 micrometer. Let's calculate how long it takes oxygen to diffuse to the center. The diffusion time is given by r squared over 2d, where d is the diffusion coefficient. For oxygen in water, d is about 2 times 10 to the negative 5 square centimeters per second. For a 1 micrometer cell, that's 10 to the negative 4 centimeters. Plugging in, we get about 0.0002 5 seconds. That's a quarter of a millisecond. Extremely fast. Now imagine we tried to make a giant cell with radius 1 millimeter. That's 1,000 times larger. The diffusion time would be 0.1 squared over 4 times 10 to the negative 5. Which is about 250 seconds. That's over 4 minutes. By the time oxygen diffused to the center, the cell would have already consumed all the oxygen near the membrane and would be suffocating. This is why large cells simply don't work. So what is the optimal cell size? Evolution has found a remarkably consistent answer. Most cells fall in the range of 1 to 100 micrometers in diameter. This isn't a coincidence. It's determined by physics. Here's a comparison. Bacteria are tiny, around 1 to 2 micrometers. Red blood cells are about 8 micrometers. Most human cells are 10 to 30 micrometers. All optimized for diffusion. You might ask, what about really long cells like neurons or muscle fibers? They can be meters long. How do they get around this constraint? The answer is, they remain thin. A neuron might be a meter long, but its axon is only a few micrometers in diameter. The critical dimension for diffusion is the radius, not the length. Additionally, neurons and other long cells have evolved active transport mechanisms. They use molecular motors to shuttle materials along their length, rather than relying on diffusion alone. There's another important equation that relates to cell size. Kleiber's law for metabolic scaling. The basal metabolic rate B equals B sub 0 times mass to the 3/4 power. This is called Kleiber's law. For individual cells, metabolism scales approximately with volume, which is mass to the 1st power. But for whole organisms, we get this 3/4 power law. This 3/4 exponent appears everywhere in biology. Heart rate, lifespan, tree height, all show this scaling. It's related to how transport networks branch. Now let's address another fascinating question. Why is it so difficult to create synthetic cells in the laboratory? The first challenge is creating a functional membrane. Cell membranes aren't just barriers, they're incredibly complex structures with thousands of proteins. A real cell membrane has a lipid bilayer studded with proteins. These proteins act as channels, pumps, receptors, and structural elements. Making this artificially is extremely difficult. The second challenge is minimum complexity. Even the simplest living cell requires hundreds of different genes and thousands of protein copies. The bacterium Mycoplasma genitalium has about 500 genes and is considered near the minimum for independent life. You can't just throw together a few molecules and get life. The third challenge is the chicken and egg problem. DNA needs proteins to replicate, but proteins need DNA to be made. How do you bootstrap this system? The fourth challenge is energy coupling. Cells need ATP, which requires complex molecular machines like ATP synthase. This machine alone has hundreds of parts. ATP synthase is a rotary motor that spins at about 100 rotations per second. It's incredibly sophisticated nanotechnology that took billions of years to evolve. Let's talk about why compartmentalization into cells provides such a huge advantage for life. First, cells concentrate reactants. In the open ocean, molecules are incredibly dilute. In a cell, you can pack them tightly together so reactions actually happen. Second, cells protect their contents from the environment. You can maintain different pH, salt concentrations, and exclude harmful substances. Third, cells localize reactions. You can keep incompatible reactions in different parts of the cell using organelles. This is like having different rooms in a house. Fourth, cells enable individual selection. Each cell can compete and evolve independently. Natural selection can't work on just a soup of molecules. Now let's discuss how eukaryotic cells partially solve the size problem and became larger than bacteria. Eukaryotic cells have internal membrane systems. The endoplasmic reticulum increases internal surface area. Mitochondria handle energy production. This compartmentalization allows larger size. The key insight is that internal membranes increase the effective surface area without increasing the outer radius. This partially solves the scaling problem. Mitochondria are especially important. They produce ATP locally throughout the cell, so you don't have to rely on diffusion of energy carriers over long distances. Let's examine the physics of why cells divide rather than growing indefinitely. We've established that as a cell grows, volume increases as the cube of radius, while surface area only increases as the square. This creates an imbalance. Volume is proportional to r cubed. Surface area is proportional to r squared. At some critical size, the cell simply cannot import nutrients and export wastes fast enough to sustain its metabolism. Division becomes necessary. Cell division solves this by creating two smaller cells. This resets the surface area to volume ratio back to a favorable value. Let's step back and consider the profound implications of these physical constraints on the evolution of life. First, cells are universal. Every living thing on Earth, from bacteria to blue whales, is made of cells. This isn't arbitrary. It's required by physics. Second, if you want to be large, you must be multicellular. A whale is made of trillions of cells, each of optimal size. You can't have a single giant cell. Third, biochemical complexity requires compartmentalization. You need to separate different processes in space. This led to the evolution of organelles. Fourth, once organisms get large, they need transport systems. Blood vessels, xylem, and phloem in plants. These solve the diffusion limitation at the organism level. Let's think about how multicellular organisms solve the same scaling problem at a larger scale. The circulatory system is essentially the organism's solution to the same problem. It brings the external environment close to every cell by branching into tiny capillaries. Capillaries are about 8 micrometers in diameter, just barely wider than a red blood cell. This ensures no cell is more than a few cell widths from a blood vessel. By branching extensively, the circulatory system creates a high surface area for exchange, solving the same physics problem at a different scale. Let me show you a more quantitative model for optimal cell size based on balancing metabolism and diffusion. The rate of nutrient uptake P sub n equals K sub 1 times the surface area, which is K sub 1 times 4 pi r squared. The rate of metabolic consumption, P sub out, equals K sub 2 times the volume, which is K sub 2 times 4/3 pi r cubed. For the cell to survive, nutrient uptake must be greater than or equal to metabolic consumption. This gives us K sub 1 times 4 pi r squared must be greater than or equal to K sub 2 times 4/3 pi r cubed. Solving for r, we get r must be less than or equal to 3K sub 1 over K sub 2. This gives us a maximum cell radius determined by the ratio of uptake to metabolic rate constants. When you plug in typical biological values for these constants, you get a maximum radius of about 50 micrometers, which matches real cell sizes remarkably well. Let's discuss the current state of synthetic cell research and what we've achieved so far. Scientists have successfully created simple lipid vesicles that form spontaneously. These are like soap bubbles, but made of the same lipids as cell membranes. We can insert individual proteins into these artificial membranes and get them to function. For example, we can create vesicles with working ion channels. We can trap DNA, RNA, and proteins inside these vesicles. We've even gotten simple protein synthesis to work inside artificial compartments. However, we have not yet created a truly self-replicating artificial cell that can grow, divide, and evolve. This remains one of the great challenges of synthetic biology. The problem is that even the minimal cell requires hundreds of genes working together. We're trying to reverse engineer a system that took billions of years to evolve. Here's an interesting question. Could life exist as free-floating nanomachines without cells? The answer is almost certainly no. Without compartmentalization, molecules would diffuse away and become too dilute to interact effectively. Concentration equals number of molecules over volume. In an open ocean, V is enormous, so C becomes vanishingly small. For most reactions, the rate depends on the concentration of reactants. Many reactions need both reactants, so the rate goes as concentration squared. The reaction rate is proportional to C squared, which equals N over V quantity squared. If V is huge, the rate becomes negligibly slow. Cells solve this problem by confining molecules to a tiny volume. This increases concentration by a factor of millions or billions compared to open water. There's another important physical constraint we haven't discussed yet: temperature and its effect on biochemistry. Most cells function in a relatively narrow temperature range, from about 0 to 50 degrees Celsius. Some extremophiles can handle higher, but it's difficult. Reaction rates follow the Arrhenius equation. K equals A times e to the negative E sub A over RT, where E sub A is activation energy and T is temperature. Higher temperatures increase reaction rates exponentially, but there's a problem. Proteins denature at high temperatures. Most proteins lose their structure and stop functioning above about 60 degrees Celsius. This is why cooking an egg makes it solid. The proteins denature. This creates an optimal temperature window. Too cold and reactions are too slow. Too hot and proteins fall apart. Most of life exists in this Goldilocks zone. Let me summarize the key points about why physics dictates that life must be cellular. First, the surface area to volume ratio, which scales as 1 over r, fundamentally constrains how large a single cell can be. Second, diffusion, which goes as r squared over d, limits how quickly molecules can move within cells. Third, compartmentalization into cells concentrates reactants, making biochemical reactions feasible. Fourth, these constraints lead to an optimal cell size range of about 1 to 100 micrometers for most organisms. Fifth, creating synthetic cells is incredibly difficult because even minimal life requires hundreds of components working together. And finally, evolution cannot escape these physical laws. Any life form in the universe would likely face similar constraints and solutions.